Hello dear friends, I Dr. Shamsher Singh welcome you all on my YouTube channel Pharma Learning. Today I will discuss with you an very novel and useful experiment in which you can use a zebrafish, and in this today experiment I will discuss the two part of that experiment. First, that how you can induce the stress in zebrafish, that mean how you can induce anxiety like behavior in a zebrafish, and the second thing, if you induce, how you can analyze the effectiveness or efficacy of your drug which can act as a anti-anxiety drug. So dear friends, this is, you can say, whole experiment. Firstly you can write down the aim of experiment, and before then that I will discuss with you that what is anxiety. So anxiety is a fear like condition, that mean whenever any person, whenever any animal or any aquatic animal like zebrafish experience anxiety or experience a fear which is associated with the anxiety like condition. So anxiety is of two types, that mean acute anxiety and a chronic anxiety. So what are the cause of anxiety in animals? Because anxiety can be analyzed in rodents as well as in aquatic animal at the experimental level in pharmacological laboratory. So what I can say, that if you want to induce anxiety in a zebrafish, what are the different models I will discuss with you. Then how we induce anxiety by using this mirror chamber apparatus I will discuss with you. And what are the cause of anxiety. The first cause of anxiety is, then whenever you will say you expose your zebrafish toward predator, that mean if you are exposing your fish toward unknown stimuli, that can cause anxiety, the first thing. The second thing, you are providing a such type of environment to your zebrafish. Suppose I will tell you the normal temperature of the water where you have placed the zebrafish should be around 25 degrees centigrade plus minus 2 degrees centigrade. But the second thing, the humidity range should be around 50 to 60%. And the third thing, there should be a maintained dark and light cycle. The light cycle and dark cycle is divided into 14 and 10 hours. So that should be maintained, so that these are the physical stimuli which can responsible for the anxiety. Now second thing, if you want to induce anxiety by using some physical stimuli or by using some instruments, how fish feel anxiety, how the rodents feel anxiety. So I will today discuss about the zebrafish which is called as a denio rario. In this fish I will discuss the first thing, what are the similarities of the zebrafish with the human being. In case of human being, you know that we are having a hypothalamic pituitary axis, that means whenever our hypothalamus is activated, it activates the pituitary gland, and that pituitary gland produce a adrenocorticotropic hormone. That adrenocorticotropic hormone activate the adrenal gland, and activation of the adrenal gland cause increase in the adrenaline secretion. Now whenever there will be an increase in the adrenaline secretion, on the other side there will be a high level of cortisones, that means cortisols. You know that adrenaline increase the sodium water retention and increase the blood pressure. The second thing, cortisol directly cause sodium water retention and thereby produce a anxiety like behavior in the animal. So that mean this HPA axis in human being is quite similar to HPR axis, that mean hypothalamic pituitary interrenal axis, which is quite similar likely in the human being. That mean whenever you will cause stress to the fish, there will be activation of the hypothalamus which activate pituitary gland, which release pituitary hormone like adrenocorticotropic hormone, which activate the renal glands. So that means this is the quite similar mechanism of the anxiety in fish as likely you have seen in the human being, or as likely you can see in some rodents. Now today, what is the aim of this experiments? The first aim, that how you can induce anxiety like behavior in the zebrafish. That means how you can induce acute anxiety, how you can induce a chronic anxiety. So that means this is the mirror chamber apparatus. This is a laptop which contain a software animes, and this laptop software is connected to a webcam, so that you can see here that our webcam is directly targeted at this site, and it is analyzing the movement of the zebrafish in a one of the apparatus which is called as a mirror chamber apparatus. So if I will talk about the first aim, to induce a acute and chronic anxiety in a zebrafish using a mirror chamber apparatus. So this is the aim of the practical. What will be the requirement? The requirement, you will need a this apparatus which is internally lined with a, you can say, mirror. It is called as a mirror chamber apparatus. The second thing, you need a laptop which should install your anime software, and this anime should be connected to the third part, webcam. So these are the three things which are required, and the fourth thing is your animal, that is zebrafish. Now I will firstly talk about what is the procedure, that how you can produce a acute model of anxiety. In case of acute model, firstly you will take your apparatus, fill up to 80% with water. The temperature of water should be 25 and plus minus 2 degrees centigrade, and you can place individual fish, or it is depends upon your practical scenario that you are using two fish, three fish, or a one fish. Basically it is better that if you want to induce anxiety, you can place a one by one fish, so that you can easily analyze everything, every parameter. So basically you can see here I have placed here 3 to 4 fish, and which are analyzed here by the webcam through the anime software. Now firstly, if you want to induce a acute anxiety, in case of acute anxiety, the total time interval of anxiety should be 4 to 7 days. What you will do, in firstly you will habituate the species zebrafish here for 3 days. After habituation, you will start your induction of anxiety. For acute, you will do a 5-minute daily trial of this. You will place a zebrafish in this water that is having a mirror chamber apparatus, and for 5 minutes daily you will place the zebrafish, and you will keep under surveillance by using your webcam. Now this protocol would be 4 to 7 days. Now what will be happen here? This is a mirror chamber apparatus. Whenever you will keep a zebrafish outside from the inner side, it is lined with a mirror. So zebrafish will easily find that there are some more fishes here. So this is a predator like condition. जैसे कि अगर आप देखें कि जेब्राफिश को ये लगेगा कि मेरे यहाँ पे और बहुत सी ऐसी फिशेज हैं विच आर लाइकली बी लुकिंग लाइक मी. So this is the fear like condition. That means this will either cause zebrafish to become aggressive, this will either cause zebrafish to become a fearful, or this will either reduce the behavior or either alter the behavior of the zebrafish. Now this model, if you want to induce, you will put the fish here for at least 7 days. You can for 4 days or a 7 days, daily for 5 minutes, and afterward you will find out what you will calculate. In case of chronic, you will place the zebrafish for 14 days daily for 5 minutes. So what you will conclude, what the parameter you will check for analyzing the level of anxiety? First thing, you will check the movement of the fish. You will keep this box into the two, you can say, division. You will keep here a one line, you draw one line, and you will check how much time the fish will stay on the upper zone. Basically in case of anxiety, the more time by the fish will be spent in the bottom zone. The first thing, you parameter you will calculate. For how long you will calculate? For 3 to 5 minutes you will calculate what was the time spent in the bottom zone, the first thing. The second thing, what was the movement of the zebrafish? Either the movement was in the central part of this beaker, or either the movement was due toward the wall of this beaker. Because whenever fish undergo anxiety, fish want to strike with the stones. जैसे हम लेक्स में देखें तो स्टोन के साथ स्ट्राइक करती हैं, ये साइन ऑफ एंजाइटी होता है. ऐसे अगर वॉल के साथ वो स्ट्राइक कर रही है, दिस इज ए साइन ऑफ एंजाइटी. द फर्स्ट थिंग यू विल कैलकुलेट द टाइम स्पेंट इन द बॉटम जोन. द सेकेंड थिंग, you will find how many time a fish strike with this particular wall of the apparatus. The third thing, how many time the fish move up and down along the wall of the apparatus. If fish is moving fast along the wall of the apparatus, this is a sign of anxiety. The fourth thing you can see, fish will not intake the food. This is the fourth sign. And the fifth sign, you will take the gasping. The gasping is the condition where the fish will travel on the upper side of the water, that means on the top layer of the water, and open his mouth. The opening of mouth is a sign that fish is feeling a hypoxia. So these are the four parameters you will calculate while inducing anxiety like behavior: acute and chronic. Acute for 7 days, chronic for 14 days in zebrafish, by four parameter. I will again explain. First thing, you will check the movement of the fish at the bottom zone at least for 5 minutes. The second thing, you will check the striking behavior of the fish with the wall. The third thing, you will check the movement of the fish from bottom to top along the wall side. The fourth thing, you can check the diet intake. And the fifth thing, you can check the gasping behavior, that means opening mouth, due to fish will feel hypoxia. So dear friend, these are the two models: acute model of anxiety and chronic model of anxiety. Now if you want to do another experiment, this will be another experiment that you can perform at B Form, M Form, PhD research level. That means if you want to check the anti-anxiety effect of your drug, any drug, if you are in PhD, you want to check the effectiveness of your drug. अगर आप एम फॉर्म में, you are doing simply a practical by using a control as well as some anxiety disease fish. Now in this case, what will be the aim? The aim of your experiment is to check the anti-anxiety effect of the drug, any drug which you want to use, in case of the zebrafish. Now in case of M form B form experiment, we will divide the zebrafish into two types. That means one will be a control zebrafish, second will be a disease zebrafish. To control zebrafish, you will not induce any anxiety, you will not provide any kind of physical stimuli. But in case of in case of disease, firstly you will induce anxiety, and then you will check your effectiveness of the drug. Now how you continue your procedure, how you proceed this your experiment? Firstly, you will take a healthy zebrafish from your tank side, and second thing, fill this mirror chamber apparatus or any apparatus which you have made up of glass with water. The third thing, keep the temperature of the water 25 degree plus minus 2 degree centigrade. And the fourth thing, now you will firstly either put the normal fish, and in case of normal fish, you will check the four things, four parameter. Firstly, swimming behavior for 5 minutes in the top zone or in the bottom zone, first thing. The second thing, you will check the striking behavior of the fish toward the wall in case of normal group. The third thing, you will check the diet intake. The fourth thing, you will check the movement of the fish across the wall, either upward and downward movement. And the fifth thing, you will check the gasping behavior. So this is for the normal group. Same you will proceed for the treatment group, in which you will firstly induce anxiety, then after anxiety you will put some treatment here. And in case of treatment, अब आप फर्स्ट पहले you will induce the anxiety, after few minutes you will give treatment, and in the treatment you will see if the during anxiety the fish movement was at the bottom side, whether the movement are increased toward the top side or not, the first parameter. The second parameter, your drug reduce the striking behavior of fish or not, the second parameter. And the third parameter, you can check that whether fish show more gasping type of behavior or not. The fourth parameter, the fish mostly in anxiety move across the wall, and if it move up and down in the central side of your this beaker, then this is the another parameter. So in this case, you will draw a table. Firstly will be the serial number, secondly will be your fishes, control fish and your disease fish. The third will be the parameter. First parameter, bottom movement and top movement. Second parameter, gasping behavior. Third parameter, striking of fish toward the wall side. And fourth parameter will be a diet intake and gasping behavior. So this will you will find for a 5 minute. So this is the practical which you can perform in M-Form. Suppose if anybody is doing a PhD work in which he is or she is using a model of zebrafish, then what you can do? Either you are taking a normal group, disease group, intervention group, and standard group. Accordingly, you divide the fish. You will take a 10 to 12 fish or what you want in a normal group, 10 to 12 fish in a disease group, 10 to 12 fish in a treatment group, and 10 to 12 in a standard group. So again you will perform all these parameter कि जो आपका नॉर्मल होगा दैट विल बी ए नो एनजाइटी, जो आपका डिजीज़ होगा उसमें पहले आप एनजाइटी इंड्यूस करेंगे, वो आपने चूज़ कर लेना. यू विल watch and you will listen very carefully so that you can induce either acute or a chronic model of anxiety. तो मैंने इससे पहले भी in the same channel I have discussed about the zebrafish and rodents. You will completely watch, like and subscribe this pharma learning so that you can understand these procedure. So in these different groups, in disease you will induce anxiety, and in treatment you will induce anxiety and give the treatment later on. If it is pre post treatment, in pre you will give a first treatment, then induce anxiety, and fourth will be your standard group. In that case you will give your standard drug. So this is the protocol. Today I have discussed with you how you can induce anxiety using a mirror chamber apparatus, and this experiment is very useful for the neurobiology as well as those students who are working in the neurological area. So you like, subscribe this channel. You all are welcome. Thank you very much.